Mitochondrial changes play important roles in human oral cancers. This protocol establishes an effective platform to isolate and purify mitochondria from human oral cancer and control tissues for large-scale proteomics analysis. Mitochondria are the centers of energy metabolism, cell signaling, and oxidative stress. Insights into mitochondrial proteome changes in oral cancers benefit our understanding molecular mechanism and the discovery of effective biomarkers and therapeutic targets. This technique uses differential-speed centrifugation in combination with density-gradient centrifugation to separate mitochondria from human oral cancer and control tissues, which results in high-quality mitochondrial samples for quantitative proteomics analysis.
The presented mitochondrial preparation coupled with ultra-Q quantitative proteomics have been successfully used in the analysis of human ovarian cancer tissue, which easily translates to analyze other tissue mitochondrial proteomes. Those who have never performed this method may find that it can be difficult to isolate mitochondria from ovarian control than cancer tissues. It's necessary to add an traction to improve control tissue homogenization and add an extra layer of density gradient medium to improve mitochondrial separation.
To begin this procedure, prepare 250 milliliters of the mitochondrial isolation buffer.